Hello, everyone, and welcome to today's webinar, Accelerating Protein Purifications with Parallelized Automated Workflows. I am Tony Biasudo, and I'll be your moderator for today's event. I am Cytiva's ACTA and chromatography specialist for the southwest of the UK, which includes Oxford, which is where today's speaker is based as part of Evotec. Today's educational webinar is presented by LabRoots and brought to you by Cytiva. So if you want to learn more, please visit cytiva.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box with the little question mark on the left, and just click Submit. You may also use the Ask a Question box to let us know if you're having any troubles seeing or hearing the presentation. And now I'd like to introduce our speaker for today, is Karthik Rajasekar, and he'll be presenting accelerating protein purifications with parallelized automated workflows. Karthik, over to you.
Thanks, Tony. Hi, all. Thanks for joining the webinar. I'm Karthik. I'm a team leader here in Protein Sciences at Evotec. I've been in Evotec for just over 6 years. And before joining Evotec, I did a PhD and a couple of postdocs where I was working on protein switches, looking at the protein biochemistry, structure biology, and also characterizing protein-protein interactions. Today, I'll be talking about accelerating protein purification with parallelized automated workflows. And during this talk, we'll start by talking about Evotec and Protein Sciences, what we do here. And then I'd move on to my talk where we first talk about automated multi-step purification. We are talking about large-scale production here. And then evaluation of protein quality and how we automate that within Evotec. And then high-throughput protein purification at small scale and how routinely we go about doing it. So first, about Evotec. Evotec is a global life sciences company, and we have platforms and technologies that can deliver precision and efficiency in drug discovery. We are spread around US and Europe and the UK. And this building here is where I am. It's in Abingdon in UK, just south of Oxford. Protein Sciences is one of the departments within Evotec, and it's split into 3 parts. Structure biology, where we try to accelerate the DMTA cycle by identifying and improving molecular interactions. Protein biochemistry, it's a department that I'm based in, and we have an efficient gene to protein pipeline here. And cell sciences, where we tailor make cellular reagents. So this is what we do and how we do it. How we work is we try to work as an extension of our partner's lab, where our scientists are driving the projects with the motivation to be better than the previous day. Protein Sciences, we've got about 120 scientists, and about 65% of them have a PhD level qualification. And the proteins that we make are used for a variety of purposes. They could be used for structure biology, like X-ray, cryo-EM, for SPR, NMR, in vivo studies, and biochemistry, and also antibody work. And we need to make protein that is fit for purpose, the right quality, purity, and the right amounts that is suitable for these experiments. And also one has to consider post-translation modifications and being in the right state that is physiologically relevant. So we need to think about the right expression host, whether it's E. coli, insect, mammalian, or yeast. And we need to think about the right construct design. And we go back through literature, homology modeling. We also have our smart in-house construct design tools to deliver on this. And once we've expressed the protein, we try to make sure that we have the right material before we scale up. The goal really is to apply rapid automated small-scale production to inform large-scale productions. Large-scale production is expensive, so you need to fail fast. You need to understand which conditions are the best conditions to scale up. So we make, even in our small quantities, we have enough protein to do a variety of QC experiments to look at the identity of the protein by using mass spec, for example, look at the oligomeric status by using analytical size exclusion or FSEC. I'll go through this today. We can look at binding by SPR, activity using plate-based experiments, stability by thermal melting using nano-DSF, for example, and we can check various buffer compositions and so on. So this is how we go through the screening strategy. So that's in brief what we do within protein sciences in Evotec. Now moving into the main part of the talk, which you're quite excited to hear, I presume, the automated multi-step purifications. So before I start this talk, I would like to have a poll question here. I assume most of you have experience working with proteins, purifying proteins in the lab. So consider a situation where you have to purify 4 different proteins, each from a 5-liter expression. So it could be from any of the expression nodes, doesn't matter. And you have to purify them using chromatographic methods. So not gravity purification, using IMAC or any affinity purification, followed by a cleavage step, and then a subtractive to remove the tag and any uncleaved protein, followed by a sec, and then alicot and freeze, finishing the complete purification. And you're doing it for 4 different proteins. How many hours do you think you would need for this exercise? So I'd give another 30 seconds or so for you to fill in your answers. It's good to see the answers are coming in really fast. About 50% of the attendees have already answered. There are no new answers in the last 5 seconds, so we'll stop the poll here. And I don't know if you could see the results of this poll. Yes, I guess now you should be able to see the results of this poll, and you could see majority of you voted it takes more than 40 hours. So with this in mind, let's go into today's talk and then try to revisit this question at the end of the presentation. So what I'm going to be presenting on the automated multi-step purification, we have split it into 4 parts here. A tandem IMAX sec, which would be a starting step to automate your purification. Then we can go and just think about tandem IMAC with on-column cleavage, and then a tandem affinity desalt, and then finally a complex multi-step purification where you have IMAC, ion exchange sec. So let's start with the simple stuff, the tandem IMAC sec. So what you need to do here is an IMAC, which is an affinity step, where you have your lysate, you load on the IMAC column using an ACTA instrument, you wash, and then you need to elute, and this elution goes automatically into the sec. And we use something called peak to loop, which I'll discuss a bit in the next few slides. Just some logistics about understanding the slides. I assume most of you have used the ACTA instrument and you're familiar with the Unicorn software. I wouldn't go too much into details of the methods in here, but during the course of this talk, you would see a lot of slides with these snips, which would be either directly from the Unicorn methods program or these sort of strips, which will also be familiar, which are blocks that you see within the Unicorn methods. So in a tandem IMAX sec, you can use it on your standard ACTA Pure instrument. There's no additional hardware necessary. All you need to do is a little tubing that would connect your outlet valve to the injection valve. And that's the only additional step that you need to do before you use any of these programs. And remember to remove it after the end of your programs. And this is what we call the peak to loop. So you have the connection done, and when you're running your program, you select the option, the peak to loop, by which your elution from your first step is now going to go directly be loaded into a loop, which can then be subsequently used by a subsequent program to inject into the next step. Okay, so and how does this method look like? So as I told you, most of you would be familiar with these blocks. So what we tend to do in this case, you could do a simple case scenario would be to do an IMAC, a single IMAC and a single sec. But because once you've automated, you could do these runs in parallel. So if you have a column val, which can take up to 5 columns, then you could have 4 IMAC columns, and the 5th one could be a sec column. So in which case you would run 4 runs in parallel. And how we like to do is that we want to make sure we load and wash each of these lysates, because you don't want your lysates sitting around for too long. So first, in the first block here, what you're doing is you equilibrate your column, you do the sample application where your lysate is loaded onto the column, and you immediately wash it. So you've already done a one-step purification. Your protein's still on the column, but it's clear of all the impurities from the lysate. You would then repeat it 4 times for all your 4 lysates. And then you move on to the IMAC elution program. We would always auto zero, remember that, because you could be slightly off after your load and wash. And followed by that, and especially if you're running multiple samples, there is ways by which you can add sample ID, so you know which sample is what, and then you would elute. And this is where you use the, as shown in the previous picture, you would use the peak to loop feature. And this elution could be a 2 ml elution that can go into a sample loop, could be a 5 ml elution depending on the column size, or a larger 10 ml elution which could go into a super loop if needed. And that could then be loaded into a sec. And you could have the sec column pre-equilibrated. If not, if this process is happening overnight, the sec column could be equilibrated, and then one after the other, each sample would be applied, and you would elute and you would complete your run. So if you're running only, as I mentioned before, only one sample, then the elution step can be part of the same program. You could have a single program with a long load wash elution sec. But if you're doing multiple programs, then it'll be good to use a scouting tool where you use this 4 times using the scouting tool. And here's an example of how this run would look like in real time. So we have in this is a 2 protein example. So you have the first protein that's loading onto the column and washing, followed by the elution, which then goes into peak loop. And if you notice carefully, we use a step gradient here instead of a linear gradient, because we have washed your protein enough, and we would like all of this material that's eluting here to go into our sample loop. So you do a step where you're basically washing everything from your column into the sample loop, and that can in turn be injected into the sec. And these 2 runs were performed one after the other automatically, and both of them performed well. And you can see both the proteins behave fairly well. You have a single nice peak in target Y, and target X, you can separate the multimeric state from the monomeric state. So next question is tandem IMAC sec with on-column cleavage. So here, just to reiterate, so now we are familiar with the IMAC load and wash step. What we would need to do is add a protease injection, an overnight incubation, and then do the IMAC wash. In this case, the protein is cleaved, so it would then load. We would use the peak to loop that we explained in the previous slides, by which you load your wash into the loop, and then you have your elutions if you want to collect them, and then your cleaved protein will be loaded onto the sec. So here's the method. How do we do this? And again, you can do this for one run, or you can do for multiple runs, up to 4 with a single column valve. IMAC load and wash on the first step, and this is very similar to what you saw on the previous slide. You have the equilibration, sample application, and the column wash. Then you have the protease injection and the overnight incubation. So what we do, we have a pre-made protease solution that is enough to fill in our column volume. So I'll show in this next slide how the column is set up, and we do it as a sample application into our system. So in this case, you can run 2 samples, so you can use the remaining sample valves to load your protease samples. And then you have a waiting period. You can use, it's not shown in here, you have the miscellaneous feature by which you can pause your runs. So you can have a period of 8 hours or 12 hours or however long you feel your protease needs to act, so the system doesn't do anything and wait. And then you then go on to the wash step, again auto zero before you have any of these proteins eluting. Then you have, in this case, the protein is in the wash because it's cleaved off the column. You then load it into peak to loop 1. And you can also then wash, if you would like to do a second wash as well, you can collect that as well, and that can go, all of them can go into the sec runs automatically. So how we set this up in the experiments, so here we've got 2 columns. So you can have 2 cases here. Your protease could be the same tag as your protein of purification, which could be his tag, or it could have a different tag, example a his for a his tag target, you could have a GST tagged protease. So what we do in the first instance, the first is the affinity step. So let's assume you have a his tagged protein, you have a his column here, and you have a GST tagged protease, you have a GST column here. You would load, and I'm following the right box here first. You would attach both the columns before the start of the method. You would load double column volume in your methods. So one column volume would actually be 2 ml because you're working with 2 columns here. And this is the actual most efficient cleavage strategy. You'd have a free protease in the mobile phase in this column, and that will be any excess protease will be caught in here. And you incubate, and after you're done, you then continue with your wash. All the protease will be subtracted here, and your cleaved protein will run through that you can collect. If you have to work with the same tag on the protease as your protein, this is slightly less efficient, but it still can be done. What we do in those cases is we only start with the first column and we do the affinity step, and then you can pause your run and then add your additional column so that you can then cleave off, catch your excess protease through here. And always consider the protease concentration and the target expression yield. So before you start your run, you need to have an idea of your expression levels, how well your protein is expressing, what volume of culture you're using to purify, so that you have a rough idea of the yield, so you can work on all of these accordingly. So then we go on to the method queue. This is the example that I was saying. You can add a wait step on either in the method queue, or you can also, using the miscellaneous, as I was mentioning in the previous slide, add it as part of your method to add this pause step. In this case, it's a 15-hour incubation. So how does this run look like in real time? So in this case, we have the iMac load and wash for the first column. So you could see all the impurities washing out during the sample application process. And then we have the protease injection and the overnight incubation. So any excess protease you could see is flowing off, and we incubate, we pause for a long period of time, and then we have the next morning automatically do 2 washes straight into the loop. So that's the first wash where my cleaved protein is running into loop 1, and then we have a second wash where any remaining leftover protein it now goes into loop 2. Of course, you're going to have any uncleaved protein and your protease coming off in the imidazole elution, which you can fractionate. And then the sample goes into sec, and there's your nice sec peak. This is now a cleaved sample running through a sec overnight untouched. On column, so just showing them with the gels. So we've seen the traces, and you could see the sample that's come off from the first sec, and on the western blot, there is no traces of your tag. It's fully cleaved. Whereas here you could see the tag giving a western. So this is an anti-his western blot. And for the second peak, again, there was a small peak. You could see small amount of protein eluting, and again, the cleavage is complete. And sec3 was our uncleaved protein, and you could see that gives us western, and any protease used that was also collected here, fully separated. So you could see now we've done one run, automated, where the protein was cleaved on the column and eluted, and we've collected the sample the next morning. Now moving on to slightly a different workflow, where we do the tandem affinity and desalting. So this is quite popular with our antibody workflow. So the difference being is we will use a versatile valve here. So instead of using a peak to loop scenario where you're eluting your protein straight into the loop, here we use a versatile valve to decide to direct the protein where it needs to go. So in this case, we're using an Acta Pure with a versatile valve, which has 4 positions, and we have 2 UV detectors. You'll figure out why that's interesting. This is, for example, a map screening pathway. Up to 10 purifications can be performed in a day. So the format, we could have the map select prism A column, 1 ml resin. If you're using 1 ml resin, we can then output of that, this could go into a desalting column or could go into a sec column. Or if you're using a larger resin 5 ml column, then that could go into a desalting directly. How does this work? So here's our Acta instrument, and we have the map resin, which is these 1 ml columns connected here, and the output of that goes into the first UV detector and then into the versatile valve. So based on the UV detection, the versatile valve, the decision is made where if the UV is below the threshold, we're not interested in that protein, it goes to the fraction collector, we collect it. If the UV is above the threshold, then we direct it to the desalting sec, and then it goes into the second UV detector, by which then we can fractionate to see where your protein of interest is. So it is an automated two-step purification. The advantage is that you can have larger volumes. You can make decision on which fraction. You don't have to predefine these are the volumes that needs to be collected. You can decide based on as it elutes where the peak starts, you can then start eluting. That's quite a useful feature. Again, with respect to programs, how does it look? The method, you would have the wash step. You're now familiar with the method settings, the equilibration of the column, the samples applied, and then the column is washed. And then to send from the first column to the second column, we have the peak to DS, which can be a desalt or a sec. We wash, and then based on the versatile valve, it makes a decision to go to the elution step. And how does this look? Here is our map select prism A chromatography resin, load and the wash. You could see all the impurities are washed off, comes through the flow through straight away. And then we have the peak to DS step where you see the peak of interest come out. Next part of this talk is the full purification where you have an IMAC, ion exchange, and sec. So in this case, we are familiar how we do the IMAC load and wash. We are familiar how we elute the protein from the IMAC, peak the loop, and that can go into the ion exchange, and then going into the sec. So when do we consider these sort of complex purifications? Any well-expressed protein greater than 2 mg per liter of expression can be used, especially when it's not aggregated target that doesn't aggregate. It's quite often. We routinely use it for resupply, so we know very well the target behavior, especially as it runs on ion exchange resin. We know what sort of salt concentration is needed to elute. High throughput, middle to large scale purification is what we would use this for. So the format, let's say we start with a 5 ml IMAC column, followed by a 5 ml ion exchange, and then sec 20. This is the 320 ml, the large sec column. The way this would happen is you have the IMAC load and the wash, followed by the elution. And from the elution, I'll show you what we do to get into the ion exchange, and then we do peak to loop. So we can, as I told you, this is easier to write a method when you know when your protein's eluting in the ion exchange resin. But what we can also do is we can then, you are now familiar with the IMAC load and wash, so I won't go too much in detail of this, and the IMAC elution. We just use a standard elution in this case. It's not a peak to loop, and we collect it in a vessel, which we will use to dilute the sodium chloride. I'll show you in the pictures next page. That would make it much more cleaner. And then we start the ion exchange. And once we load the ion exchange and we elute them in 3 parts of 1/3 salt concentration. And each of these elutions will go into a peak to loop situation. So even if you know where your peak elutes, then you can have your gradient accordingly set up so that you will have your peak in the first elution. So you collect it, then that would go into the sec. But if you don't know where your peak is going to elute, then you can at least do a 3-step elution so that you have divided your elution into 3 parts and collect them all separately in peak to loop, loading into the sec. So how is this set up? So the important part is you have the sample inlet from the ion exchange and the outlet from the IMAC elution is into the same vessel. So what we do is we have the dilution buffer in a Duran bottle. The IMAC elution goes into the same bottle. Your sample gets diluted so that your salt concentration gets lower, and then the ion exchange is taken from the same bottle. And you can do all the calculations to work out exactly how much volume you need for this, for the dilution buffer you need in the sample inlet. So as I told you, we have a 3-step elution process. So in this, we equilibrate the ion exchange, we have the sample application, which is straight from the Duran bottle I showed you in the previous page, and then the column is washed, and then we incubate it in 3 steps. So we incubate with the increasing salt concentration. So we are doing step gradients here. We incubate with about 0.9 column volumes of your buffer. We do a 10-minute incubation, and then the elution goes now. We do a 3-column volume elution that now goes into peak to loop. So in our first loop, we've got 33% in this example of salt being collected. We know in this case specifically that's where our protein should elute. So you have the IMAC load and wash example and the IMAC elution, and then you have the ion exchange elution. So as I was telling you, we'll do 3 different steps here: a 10-minute incubation and a loop wash, and 10-minute incubation and a loop wash. So that's your initial wash. You do the loop wash, your loop is clean, and then the first 33% of the salt gets eluted as a peak. That's your first peak to loop. And then that goes to loop 1. And then you clean your second loop as well. You incubate 10 minutes with your increased salt buffer, and then you elute it. And then you go up to the third concentration, and then you collect your third sample as well. As you can see, majority of our protein eluted in our first loop, and there's hardly any protein in the other 2. This is just a test case scenario. So if you had multiple peaks eluting and increasing salt concentration, we would have collected all of them into 3 different loops that we can subsequently run in a sec. So this is the first loop running in a sec, and you can see a really nice peak. So on the right, and the salt is separated on the sec column. On the right, we have the STS page of the entire purification. So that's your total lane. And in the solubilized fraction, you can see this is a very well overexpressed protein, but there are quite a few impurities. You can see the level of impurity in here, whereas after the IMAC ion exchange sec, we've got a clean protein eluting here. So, and that purification is untouched from the process of starting the IMAC till the end. So just to sum, next we would go into the protein evaluation of protein quality. But before we go into that, I just want to go back into the pool. And now just to summarize what we've learned so far, the first step is a automated IMAC sec, which is a 2-step process, fairly straightforward. All we need to do is just do the connection as mentioned in that slide, and we can do the peak to loop function. And you can not just do 1 run, you can parallelize it up to 4 runs with a single column valve. And moving on from there, you can do an automated IMAC cleavage sec using the same principles, and you can slightly adapt it using suitable vessels to collect your elutions from the IMAC so that it can be used as a load into the ion exchange. And so then you can do an IMAC ion exchange sec, something more complicated, right? So if you've understood the concept of automating multi-step and also doing parallel purifications, now based on what we have understood, if I can request all of you to vote again, it's the same question as before. Based on today's discussion, using ACT automation, how many hours would you need to purify again? It's the same scenario, 4 different overexpressed proteins from 5 liters using IMAC cleavage, subtractive IMAC, and sec. So can I request you to vote now? Sorry, do I have to start the voting? Thank you. We're still getting responses, so I'll give another 5 more seconds. Brilliant. So there are no new responses in the last few seconds. So yeah, you could see, I'm really pleased with the answer to see that now it's skewed back to 20 to 30 hours. If we plan a little bit more, we could even do with this under 20 hours, but without breaking too much sweat. I think with the right automation in place, we can do 4 proteins purified from a 5-liter pellet easily between 20 and 30 hours. So thanks very much to everyone for voting. So moving on, let's now go to evaluation of protein quality. So There are a couple of scenarios when you need to evaluate protein quality. So one, before even you start your large scale, when you're doing your small scale expression trials, you need to know, you can do gels and Westerns to see the purity of your protein. You can confirm the identity using Western or mass spec. But what is crucial is to see how aggregated your protein is. You need a single nice homogeneous material that is non-aggregated. Apologies. And to confirm that, we use an analytical sizing column, and to increase the sensitivity, we have it attached to a fluorescence detector. So that's what we call as an FSEC instrument. So the instrument setup is shown here. So you can use either an Acta Go or an Acta Pure. If you're using columns that needs pressures more than 5 megapascals, then you would go for the Pure. But for most of the standard Cytiva columns, Go would be ideal. We have the autosampler, and we also have the fluorescence detector. So what we routinely do is most of the proteins, as you're familiar, you're working with his tagged proteins, and there is a way to attach a fluorescent tag into your his tagged proteins. And this is based on a publication. What we use is a strep tag peptide that's got a fluorescein on it, and it's got cysteines through which nickel NTA is attached. So this can then be coordinated to nickel, which means then it would bind to any his tagged protein. And we have this prepared in-house, and we can attach it to our target of interest. So even if your target is in a lysate, this would bind specifically to your target. Thereby, you can identify your protein in a crude lysate. If you have a purified material but really low concentration protein, typically seen in membrane proteins, for example, you can then attach this as well to follow your protein. So we've been through the instrument setup and how we detect our protein. We have an autosampler by which we can do multiple runs in a single go. And the Acta Go's go to UV detection. As mentioned, following the UV detection, we have a fluorescence detection and a fraction collector. And when you're doing these sort of experiments, you need to make sure the resolution of your column is super good. And to get best resolution from an instrument, the tubing size is crucial. The thinner the tubing you go, the better the resolution. So here on the left panel, you have the green tubing, which is 0.75 mm dia, and compared to the blue tubing, which is 0.25 mm dia. And you could see the peak shapes already, and for a standard run here. So the improvement in resolution might look marginal, but this actually makes difference when you're purifying your protein and helps you to separate those monomers from multimers. And in this case is an example where we are looking at a membrane protein target, different detergents and buffer conditions. We've shown some of the conditions here. This is a 36 condition scout, and where we can clearly see in various detergents how this protein behaves. And in this panel is where we expect our desired monomers with the right micelles, and we do see oligomers in here, and some of them also showing aggregates. So very quickly, so what we could do is once we've done our expression scout, we do our pull down, we take these elutions, we bind them to the FSEC probe, and we can then run them on these instruments. As I told you, we can also run them on the lysates, and your target specifically would light up and would give you a nice peak like this. And we can identify the best condition from the slew of conditions that we tried. And the final topic so we've talked about automated purification, and then we talked about assessing the protein quality, specifically looking at the aggregation status. And final topic today would be the high throughput protein purification, where we are looking at small scale production. So the routine workflow that we have, so in this case, we can do 48 purifications in parallel. And these are not just one-step pull downs. These are pull downs followed by a sizing purification. So in this example, we are doing 50 ml growths. We do always do some backup growths, and we lyse them. And then we do an automated one-step pull down using a magnetic pull down system. And that goes on to an automated sec. As I mentioned before, we have an auto sampler by which we can load all of this one after the other into the sec. And then we continue with the QCs where we have STS page and Westerns. And on the panel on the top, we have the molecular weight standards. And the panel at the bottom, we have all the 48 constructs, which in this case is a bioengineering project. We have got different constructs for the same target. You can see they're all yielding at the same level and with different expression concentrations in this. And then we go on to check the ID, where we do the mass spec in an automated way, and we can then look at the activity using plate-based activity screens. So in that way, we can really fasten the rate of doing small-scale purifications in a really automated and high-throughput fashion. With that, I would like to finish my presentation. So key takeaway today is you can save a lot of time with automated multi-step purification, both at the large scale and at the small scale, and doing high throughput runs. Multi-step purifications, even complicated purifications, can be automated if you have enough information about the protein, and there's a lot of the time saving is really significant. What automation also helps to deliver is it improves reproducibility and reduce manual errors in protein production. And hopefully today I've shared some of our practical workflow insights, how to evaluate protein levels and protein QCs at small scale, and also how to perform high throughput purifications. So just to put some thoughts into perspective, if we are doing manual purifications, a 5-liter IMAC affinity followed by a sec might take 2 days, around 2 days in a manual process, but in an automated way, we can do it in 1 day, with the hands-on time being much less than that. And again, a 4 times 5-liter, as we discussed before, could easily take 5 days, and we can do that with 2 days using the automated step. And 2 times IMAC cleavage, ion exchange sec, the multi-step that we discussed today. If you have to do 2 full purifications, it could take 2 weeks, which could be 10 working days. But if you automate that, you can achieve that in 3 days. So it's automation and parallelization together can definitely give a lot of savings in resources. Hope that's been very helpful to all of you. I would like to thank all the people who have contributed to this. Nicola, Claudia, Josie, Lorraine have contributed a lot on the methods development and also the data collection. Cedric and James are really interested in membrane proteins, and they are very experienced with it. So if anyone wants to contact us in future working with membrane proteins, they'll be happy to have a chat. Frank has Francesco Rao or Frank has a lot of interest in ACTA methods development and contributed to the development of the slides. And John is our head of the department. I'd also like to thank Amar, who's helped to manage to get this talk together. And from Cytiva, we work very closely with them. A lot of the development's been done in collaboration with Cytiva. We really like to thank Cytiva on this occasion, particularly Chris and Tony, Lisa, Sasita, and Soumya. And that's all from us. So thank you very much for joining today's webinar.
Thank you so much, Karthik, for that fantastic seminar. I just love to see all those beautiful automated chromatograms just one after the other. It's just fantastic. We will now start our live Q&A portion. If you have any questions, thank you to those that already submitted them. If you have any additional questions, please share them with us now. Just have to type your question into the Ask a Question box and click Submit. And we'll try to answer as many of your questions as we have time for. So just to get us started, Karthik, so can you explain a little bit? So there is a question asking to explain how the column valve will switch when you do your IMAC sec, your tandem IMAC sec. How to ensure that the column valve will switch between steps, right? So could you speak a little bit more as to how the pick-to-loop process works in a method queue?
So sorry, is it a question about column valves or the?
Yes. So so the question is, how will the column valve switch between your IMAC to your sec column in your tandem sec?
So in the method, you would establish. I'm not sure if it's actually present here. You would mention the column position when you're writing your methods. So let's say you've got 4 IMAC columns, which will be in column position 1, 2, 3, and 4, and you have 1 sec column, which will be in column position 5. So, and then you have your loop valve, and you have your sample loop. So your elution from your first IMAC column goes into the sample loop. Let's take a simple scenario where you have only 1 loop. Elution from this first column goes into the sample loop. First column is in column position 1, and then you're eluting that into the sample loop, which is then injected into the sec column, which is in position 5, as mentioned in your method. Does that answer the question, Tony?
Yes. So just to clarify, it is how, as Karthik explained, so you would make use of a method Q, and in a method Q, your methods are connected through the pick to loop, but they're independent from each other. So each method will have a new column position defined for that run. And so in your first run, you'll be using your histrap, your IMAC column. And in the subsequent step in your method queue, you will have switched that column position to the sec column, and that's how you alternate between column positions in a tandem IMAC sec. And this is explained earlier. It's just if people are unfamiliar with method queues, they might not be aware that that's intrinsic to the principle of the queue. So can I ask? So what is the effect of different expression yields when you're dealing with automated IMAX-sec? So because as you're doing peak-to-loop collection, different expression yields might result in different sort of volumes of elution. And so how do you guys deal with that variation?
So we usually do multiple runs. So once we've done our expression, we evaluate the expression levels. And usually when we're doing IMAX-sec, we try to club them in groups of 8 so that 4 can go in a single batch. So based on the expression level, so assuming we've done similar volumes of expression, we then evaluate the expression levels and then batch them together so that you could use similar sized columns, right size of elution volumes, and suitable sec column volumes as well. So if you have a large, if you're working with more than 30-40 megs, then you would probably want to go into a larger sec column, and you would batch those large expressors together and the small expressors separately. So as I mentioned before, having a rough idea of the expression levels would enable you to plan the experiment properly, and you can always batch similar expressing proteins together. And you would have this information from your scouting or even your expression trials that you might have done much earlier.
That makes sense. Thank you. And moving on to the IMAX-sec with a cleavage step in between. Have you tried more than one protease or I would guess biosimulation agent for in-column cleavage?
Yeah, so we have tried proteolysis efficiency in an automated way.
Okay.
And that's what we have done. But yes, I can imagine you can do any overnight incubation process using the same method. So it doesn't have to be a specific protease. You can try different proteases. You can try different modification enzymes, for example. As long as you can separate them, they've got different tags, you would separate them.
That's very sensible. And I suppose if you have the option of a loop valve, you can have, if you have different, if you have a need for different enzymes for different proteins, you would have the flexibility of having that option. Yeah, that makes sense. Right. So I have another question here, which is quite interesting. So for antibody purification using Prisma A, since your antibody is usually eluted at low pH, do you find that when you are doing the desalting step, as you're eluting and collecting, do you find that you see often precipitation? And do you need to neutralize your elution to a higher pH to avoid clogging the desalting column?
Yes. So on these examples that we have presented, we didn't have to neutralize, but very often where we are doing low pH elutions, neutralization is a preferred step before we go to a buffer exchange.
Yeah, I think it's really interesting because it can be quite a problematic situation if you're automating and you suddenly have this misbehaving construct that
I would recommend the strategy we used where for the IMAC ion exchange sec, where you elute it into a dilution buffer,
Yeah.
And then you desalt.
Yeah, I'm also thinking, so we have, Cytiva has a new mild dilution resin for antibodies, so a variant of Prisma A, which I don't know if you guys know about, where we have mild dilution conditions. And it's precisely for instances where you get these misbehaving antibodies that are unstable at low pH, usually because of aggregation, as this question highlights. You can bypass the need to go to such low pHs and just work at pH 5, for example, and get elution without aggregation. So I think it is a really interesting point, and I really appreciate that. It's a really good question. Sorry, so another question. Yeah, and this is a question that I personally have. So it's how do you handle difficult molecules or protein constructs which are not behaving in the standard way? Do you need to have pre-existing knowledge of a sample's behavior, or at least characterize the sample partially before you can implement these methods, or can some of these methods be implemented blindly?
So as I mentioned, a lot of the targets that we work at Evotec are challenging. They are human drug discovery related targets, and many of them are quite challenging. So while this slide showed how easy it is to purify proteins, we fully understand purifying protein is a super challenging task, and that's why I said fail fast. We need to understand what is the best condition to scale up with as little resources as possible. So we do a small scale scouting. So when you're working with a challenging target, what we routinely do is we ought to design a lot of different constructs, and we try to express them in different expression hosts at different expression conditions, and we test them in small scale, in usually 3 to 5 ml scale. That would take less resources, and we using that expression levels, we try to do, and if it's a soluble protein, you would get enough material to do variety of these experiments. If it's a membrane protein, you might not get enough to do many of these experiments. So we try to get as much information as we can get from that small scale expression, which can then help us to decide which ones to scale up. And we always find surprises when we scale up. It might not be scalable, and that's what makes protein interesting to work with. But we fully understand there are lots of targets out there which is super challenging.
Yeah, no, and to that, there's another question here that speaks a little bit, and I'd be interested to know your experience. Did you find that these automated methods are more amenable to a certain kind of protein? So, for example, are they easier to implement for soluble proteins as opposed to membrane proteins, or is it just more target dependent and class dependent?
It depends on the question that we are trying to ask, right? So if you need to run 5 IMAX-sec purifications, of course, the soluble protein is going to be easy to handle, and it doesn't matter whether it's automated or not. Soluble proteins are easier to handle than membrane proteins. But when you're working with membrane proteins, there are much more different type of challenges. Even before we go into purification, you need to identify what's the best detergent to solubilize in. We need to get more information before we scale up, and we can use a lot of automation to answer those questions, which is very relevant for membrane proteins, like what we have shown here. And then once you identify the best condition, then you could probably do a strep sec, which could be fully automated. So then once you've done your membrane solubilization, you automatically do a strep purification followed by a sec, and in that way automation is faster than manual handling. Your membrane proteins are not very stable. You're purifying at much faster rate than doing it manually. So automation adds value in a different way. Yeah. So it's a kind of question that we want to answer, and we can adapt our automation depending on the nature of the problem.
Yeah, I think you are absolutely right in that. It may be a bit daunting to begin with, I guess, automating your methods, but there are so many benefits that you might not even be aware of. Just considering how quickly you can move on between your targets if you're in a screening campaign, for example. That you can get around some issues that are just intrinsic to manual purification. So yeah, absolutely right. So a question that I think it's about, you know, in your opinion, do you not find that peak to loop methods can be risky, um, simply because you might be sort of transferring material that is aggregated um, from your um, eluted sample, eluted sort of column, straight into SEC without having first checked that, the sample is stable, that there isn't a lot of aggregates that you can get rid of in with say different sort of molecular weight cutoffs, for example. So do you find that these speak to loop methods pose an intrinsic risk when it comes to sort of potentially transferring material that isn't, the best shape or the best aggregation state?
Just have right pre-filters before your columns and a good cleaning strategy at the end. So
Yeah,
yeah, we would, yeah, there is always going to be 1 out of 20 targets that might not behave well, and as I told you, you might have some information from your small scale test where after your in a small scale test you've done a pull down and an ASEC, for example, it's going to be an easy project. You can then automate
yeah,
them with some confidence. But if you're going in blind, yes, there is a risk.
yeah.
There's definitely a risk, and you might, the column might not be happy after that, but you need a really good cleaning regime.
That's a really, really important point, actually. Not only with automation, but just in general, it's cleaning your columns is always the best practice to try and minimize long-term complications. So yeah, it's a
And
really,
the
really good point.
cleaning protocol can also be automated, right? So you can
Yes, absolutely. Yeah, I
so
mean that is
so
that
it
is
could
the
be
best use
it
of
could be
automation, in my opinion.
it
You
could
have
be the
to
standard
tell.
runs you set up at the end of every week, where
Yeah,
it's whether it's sodium hydroxide or whatever your cleaning regime is,
whatever. Yeah, reagents.
finally into the storage condition, and that could be
Yeah,
automated as well. So after a busy week, you start a program, you have buffers pre-made, you start your program, and over the weekend your column is clean and ready.
yeah, and I think it lowers that engagement requirement because you can just set it up and walk away. I think it might end up making it easier, right, to implement cleaning protocols, which are so important and
If,
lacking in many cases. So yeah, that's really good.
if
I think yeah, go on. Sorry, apologies. Yeah,
Sorry, because you said the word daunting. If anyone felt automation is daunting, I would strongly recommend them to try. Start with a simple IMAX sec. It's really simple, and you can even run dummy runs. You don't need to spare your protein over. On a Friday afternoon, set up the
absolutely.
program. Use if you have any lysozyme in the lab, use that as your starting material and see how
Yeah,
the protein goes through. Or you can just run a blank run to see how everything
yeah,
goes. And
BSA. Yeah, absolutely. Yeah,
and,
yeah,
and, and all of these stuff, and that would give you confidence. Setting up these, there are a few points to remember. You don't want any leaks. You want all your lines to be connected before you start well, because there are multiple, it's not just one column, there are multiple columns, multiple buffer inlets. It's a lot to look into. So when you get to this step, you might be really busy. Your ACTA instrument will be super busy with so many tubings around it. So I would suggest people start here with a simple run. If you have some histacked proteins, use that as if you have a lysate, just lyse on a Friday afternoon and set this up and see what happens. And once you've started using it, once you familiarize yourself, you're confident it's not failing, then you'd be using this more than the manual purification.
yeah, yeah. And on that actually note, if anyone needs support, you can always contact your local ACTA specialist. That is um, probably the aspect of my job that I enjoy the most, which is going to someone's lab and try to helping them establish these new protocols and new strategies for purification. It's always a pleasure and a joy to sort of help someone use our ACTA to the fullest. So yeah, always feel free to reach out to your local specialist if you need support, please. So we have time for one more question, and I think it's a really interesting question and relevant question. So someone is asking if the buffer screening that you spoke about at the end. In I think if not the last slide, possibly the previous one, possibly is the membrane protein screening, buffer screening that you did. Was that automated as well? Did you automate the buffer screening as well?
Yes, the injections are automated. So we use an autosampler. It's called the Alias autosampler that's attached to the instrument. Once we've done the pull downs, which is using an automated instrument, we get the elutions. You can either take the elutions and run here, or you can also add the lysates with your the probe that I mentioned in here. So assume you have 48 different samples. You have the lysate, 48 lysates, and 48 elutions, and you can run all of them together, and in about a day you get your results.
Yeah, and I think I know that you actually have written an application note where you also discuss detergent screening using an alias, which is now available on our website. And so it's another incredible resource where you guys discuss automation in a slightly different context, but to try and streamline and fast track screening of membrane proteins with this really crucial step, which is detergent screening, which can make or break your membrane protein purification. So it's another really fantastic example of the work that you guys do at Evotec. So right, so thank you so much, Karthik. This has been a fantastic session, and just such a pleasure to speak with you again. We'd also like to thank LabRoots, Evotec, of course, and Cytiva for organizing today's webinar. We, of course, like to thank everyone who's joined. Thank you so much for participating and for your amazing questions. If we didn't get to answer your question specifically, we'll pass these on to Karthik, and we'll be able to get back to you directly, of course. And this session has been recorded, so this webinar will be available on demand through the link of your registration. So you can always come back to it if you want to take a look at the content that Karthik has shared today. And you can please share it with your colleagues, and let's try to get the word out on how to be more efficient with your purification strategy. So thank you again so much, and until next time. Goodbye.
Thank you very much. Bye.